Well, welcome everyone, and thank you for joining Amethyst at Penn's December 2024 monthly seminar. We are proud to offer this content through our Amethyst at Penn Hub, which stands for Achieving Maternal Equity and Transforming Health Through Implementation Science and Training. Funding for this hub comes from the NIH's Improve Initiative, which aims to reduce maternal mortality, improve pregnancy, perinatal, and postpartum care, and to address the factors that have led to severe disparities in outcomes for marginalized groups. As a reminder, we will be recording this discussion and it will be available on our YouTube channel for review after the session. Please feel free to put any questions in the chat, and there will also be time for additional questions at the end. This month, we're fortunate to have Dr. Alisa Stephens Shields here to speak with us about designing implementation science research studies. Dr. Stephens Shields is an associate professor in the Department of Biostatistics, Epidemiology and Informatics, and director of the Biostatistics and Data Science Core at Penn, and we're very thankful to have her speaking with us today. So with that, I will turn it over to Alisa.
Thank you so much, Casey, and thank you all for attending. I hope you find this informative, and I look forward to your discussion and questions. So please don't be bashful about interrupting with a question. So as Casey mentioned, I'll be discussing study design in implementation science. I'll mostly be talking about randomized studies because many of the studies that I work in are randomized trials. However, a lot of the same elements are equally applicable to observational studies. And one of the examples that I use is actually motivated by an observational study that I'm currently working on. So with that said, let's move on. So I'll first touch on hybrid effectiveness implementation designs and Hattrick US. The understanding scalability version will be our running example. I'll then touch on some design essentials that are really the skeleton of any study. There are certain fundamental aspects of a design. You know, what is the outcome? What is the intervention? What is the target population? Um, I'll then describe some randomized trial designs, particularly with a focus on cluster randomized trials, because they're often used in implementation studies just by the nature of the type of interventions that we're often using. But individually randomized trials are a nice starting point because they're very easy to understand. So we'll start there and then build. Um, I'll then discuss power considerations and then some key points and lessons learned. Okay. So a hybrid effectiveness implementation design is one that takes a dual focus a priori in assessing clinical effectiveness and implementation. So there's type 1 through type 3 designs, and my understanding is there's a different, I guess, formality about the way that effectiveness and implementation outcomes are considered and evaluated within these trials. So type 1s test effects of the clinical intervention on effectiveness, but they also gather information on implementation. Whereas type 2s test both the effectiveness and the implementation outcomes. And Megan, if you want to chime in here and be more precise, I totally welcome you to. Um, any
I
insight?
think I think you're good. And when we come to question and answer, perhaps we could talk a little bit more. Sometimes it's useful to specify which type, and people are certainly used to that. But I think increasingly, the boundaries between them are blurring a little
Blurred.
bit. So
That makes
sometimes you
sense.
don't have to put your nickel down and say, I'm a this or a that.
That's good to know. And then, as I mentioned, our running example will be handoffs and transitions in critical care, understanding scalability, which I have the pleasure of working on with Dr. Lane Fall and with Casey. And so this is a type 2 hybrid effectiveness implementation trial that has a stepped wedge design, consistent with the dual objectives that we observed on the previous slide for hybrid implementation studies. Is that Hattrick is looking at the effects of an implementation of a standardized handoff protocol from the OR to the ICU on both fidelity to handoff elements, and then also asking whether it reduces the rate of new onset organ failures. Um, in this stepped wedge trial, there's 12 ICUs. And they, according to the step wedge design, all start in the control state, and then one by one cross over to the intervention state. Um, Originally, we had thought we would have 6 to 8 week periods, but they got longer. So it's really more like 10 to 12 or 4 months. And that's one of the challenges that we'll discuss later. Um, the co-primary outcomes in Hattrick US are implementation, so fidelity to the protocol, which has these 10 components that the teams involved in handoffs are supposed to conduct. And so these include introductions, stabilization of the patient, huddle of clinicians, anesthesia reporting, exchange of contact information, and several others. And then the effectiveness outcome is the number of new onset organ failures. So what are the design essentials for hybrid for really any study design, but how does that change a little bit when we're looking at hybrid effectiveness implementation studies? So the essential components of designing any study, right, are to identify your research question. Here, we're typically going to have two, right, because we're going to have an implementation objective and an effectiveness objective. We then want to identify the target populations and also the units that will be drawing from those populations. And those might not be exactly the same across the two objectives. We want to precisely define our intervention or exposure levels and comparators. We want to precisely define our outcomes, and necessarily we'll have two because it's a hybrid implementation effectiveness design. Um, we then want to select an appropriate study design to answer our research questions, and we want to be able to strongly justify that design according to the intervention, according to the disease area that we're working in, according to the feasibility. Um, so all of those things go in mind, and I'd say that's the one place where they meet, right? We want to kind of reconcile the differences in the outcomes, the differences in the exposure, and the differences in the target populations. They have to come together at the point of the study design that's finally selected. We then determine the sample size to power our study so that we can reliably answer our research questions. And I note sample sizes because depending upon the level at which your implementation and your effectiveness outcomes are measured, there might be two different sample sizes. Or we might do two sample size calculations that yield different numbers and we have to pick one. Um, and then we finally will choose statistical analysis methods that match our design and ideally are efficient and unbiased, et cetera. So identify your research questions. We want to be very clear in our statement of our implementation and effectiveness objectives, right? So in Hattrick US, as we already saw, we wanted to know if implementation of a standardized handoff protocol from the OR to the ICU improved fidelity to the handoff elements and also reduced new onset organ failures, right? So looking at fidelity is our implementation objective, and new onset organ failures reflects our effectiveness objective. Next, I think it's helpful if we note that we're considering these elements in parallel across our two objectives. And so we can construct this table here and say, well, how do we think about the target population and the units for the implementation objective? And how do we think about it for the effectiveness objective? And we can go through our listing of essential design elements in this paired way. So what would that look like, for example, in Hattrick US, is that for the target population and the units that are drawn from that target population, for the implementation outcome in Hattrick US, we're looking at patient handoffs. And so there, we're considering all of the patients that are transitioned from the OR to the ICU, and we're drawing one handoff at a time to construct our analytic sample or our trial sample. For the effectiveness outcomes, this has actually varied a bit over the course of Hattrick, but I will start with where we initially conceived we would be as far as the level of the effectiveness outcomes in the target population. And so here we are considering the ICUs and the number of new onset organ failures is going to be the outcome that's assessed at each ICU. And so here we're already starting to see how we can have different levels or different units of observations in our consideration of the implementation and effectiveness objectives. The intervention or exposure levels and comparators, I would think typically the same across them just based on the nature of the study, although I will actually walk that back because I can think of another study that I'm working on where they're not quite the same. And so in Hattrick, we're looking at adoption of an ICU level tailored protocol. Sometimes the effectiveness outcome might consider some non-randomized thing kind of downstream of that, which is what I've seen in another study that I'm working on. Next, we have to define our outcomes for each objective. So in Hattrick, the number of components that are completed during the handoff is our outcome, and that's a count outcome. And when we're reporting on that count outcome, we'll likely report on a ratio of the increase or decrease in the mean number of components that are adhered to in the intervention state versus the control state. With our effectiveness outcomes, we originally thought that we would have at the level of the ICU a rate of new onset organ failures. But it might actually be the case as we're getting that data that we have individual level data on the occurrence of new onset organ failure. So that would be kind of a binary version of the outcome looking at the patient level, right? So we have all of these different kind of levels and outcomes and variable types at play when we're considering our hybrid effectiveness implementation designs. Okay, so they all meet when we select our study design by which we're going to sample individuals or practices into our study and collect data on them. And when we are selecting our study design, we want to reconcile that design across both objectives. It should work. Some of the considerations that would inform the study design that we select are whether we need to have an individually randomized or a cluster randomized study. Oftentimes, the nature of the intervention will determine whether it's appropriate to randomize individuals or if we need to randomize larger units. In Hattrick US, the intervention is conceived at the practice level because there's a number of staff that are involved and need to be trained to adopt this protocol. So it doesn't really make sense to consider it being randomized at the handoff level, which is the lower level in that study. Other interventions might consider some type of messaging to adhere to some evidence-based practice that you can send directly to the patient. So there it might be more feasible to do an individually randomized design. Whether we want to have a parallel design where units are randomized to one level of the intervention and stay in that the whole time, or whether we want to have a crossover or a stepped wedge design. And then we also need to consider if our design will have longitudinal or repeated measures. And so these are some aspects that go into selecting the experimental design that's used to address our study questions, right? So in Hattrick US, we had this tailored protocol involving multiple clinical providers, and so that led us to use a cluster randomized intervention. The tailoring of the protocol in order to create a standardized protocol at each site is a somewhat intensive procedure as it involves talking to a lot of different staff, setting up a lot of different meetings and interviews. And so based on that, the staggered rollout that is a feature of the stepped wedge cluster randomized trial design was attractive. And so Hattrick US uh, ended up having this stepped wedge cluster randomized trial design. Megan, I'll again ask if you want to chime in and say anything about the motivation for the design in Hattrick US.
Yeah, I think one of the things you have to think about when you're trying to sort out study design is sort of balancing threats to inference and internal validity with external validity and transferability or generalizability. So that's certainly one consideration.
A
Because
great
this
point.
study was based in active clinical care settings, we needed it to be pretty pragmatic. So that was one consideration. There were contamination considerations relevant to what Alisa already talked about, and so you can't, just have assign one person to doing things a particular way. Um, so that's why they're all clustered together. And then it's really hard when you're telling folks, we have a standard of care. We want you to follow it. Let's randomize you to get it or not, right? There are a lot of sites that that's not going to be acceptable to, especially in implementation research. So often in implementation research, you'll see people assign sites at different times, but they can at least guarantee every site that they will actually get the intervention. And so that's sort of where the concept of the stepped wedge came in. But certainly they are difficult to execute because meeting the assumptions of the stepped wedge when you have a behavioral intervention, as we've learned, is pretty tricky.
Yes, thank you for that more complete explanation. So then we power the study according to our selected design. So we're going to think about the sample size for each objective, an effect size for each objective, and we typically set our type 1 error to 0.05. That's kind of conventional. And then we'll choose statistical methods to match the selected design, considering the unit of analysis and observation, whether we need to use methods for cluster longitudinal data, and the sample size for the respective outcome. Um, so in Hattrick US, again, we had 12 ICUs. The implementation outcome is multi-level in the sense that we have handoffs that are nested within ICUs, and that's all repeated over time. For effectiveness, we originally envisioned that we would just have ICU level data, and then that's repeated over time. So there is some some extent of repeated measures across both objectives, but the nesting at a fixed time within a higher level is unique to the implementation objective. And for both objectives, we have an outcome that is counted as the number of new onset organ failures or the number of completed handoff elements for implementation. And so both of these are using negative mixed effect binomial regression models, but we note that there's multiple sources of correlation within the implementation objective and only one in the effectiveness objective if we consider the ICU level data. So what are some example RCT designs? So the individually randomized two-arm parallel design is probably the simplest and most familiar to everyone. And I just want to echo that we could also consider observational studies in this manner, except for the fact that the treatment allocation is not randomized. But you can imagine having a set of patients who select themselves into a treatment A or a treatment B, and then you observe them and assess their outcomes at the same time in both treatment levels. Right, so a feature of this is that all randomized units are only allocated to one level of the intervention. You get concurrent observation and assessment of the intervention and control outcomes. But some limitations of this design are that if the intervention is administered at a higher level and outcomes are assessed at a lower level, then this creates this violation of independence because you have this correlation of the lower level outcomes within the higher level intervention assigned groups, as we see in Hattrick with the implementation outcomes. And then we also could be concerned about contamination of intervention versus control among individuals who might be in the same group. So say two different individuals are randomized to different levels of the intervention, but they see the same provider, right? So that could be a source of contamination that would lead you to use a cluster design. Speaking to the disadvantages of each randomized unit only getting one level of the intervention, as Megan already mentioned, it is unappealing to potential participants to potentially be randomized to get the control. And to know that there's some experimental thing that we think is better, but that they're not going to get it. So that's disincentivizing for enrollment. And then the rollout of the intervention, because all of the units randomized to the intervention are supposed to receive it at the same time because follow-up starts at the same time for everyone in a simple parallel two-arm trial can be may not be feasible. It may not be feasible to get the intervention started at all of the sites that were randomized to it or for all of the units that were randomized to it at the same time, right? So all of these things would lead us to consider other designs. So for the first couple of challenges, as far as contamination and the nature of the intervention, we could move to a parallel cluster randomized design. And here we would randomize groups of individuals to an intervention or control. And we would still start them at the same time. Each group would be randomized to one and only receive that level of the intervention, and we would observe them in parallel. It's just that, the assignment would look like these groups of individuals getting the same thing as we see here. So additional parameters that are involved in a cluster randomized design are the intra-cluster correlation. And now we have to think about sample size in two ways, the number of cluster and the number of individuals per cluster. Some challenges that are associated with cluster randomized trials is that there's typically a smaller number of randomized units. And because of this, imbalances in characteristics across intervention and control arms are more likely. And so an implication is that we need to be really careful about enforcing balance when we allocate the groups or the clusters to a treatment or a control. And so we often use things like covariate constrained randomization, where we look at, all of the possible ways that the clusters can be allocated to intervention or control, and then we only randomize among the ones that lead to sufficient balance across the what would be the treatment and control groups. Another challenge of cluster randomized trials is that the large sample theory underlying statistical approaches may not hold. It might have been that we had 15,000 patients, but now we only have 30 clusters, right? Depending upon how many patients there are within each cluster, whether we're considering hospitals or classrooms or things of that nature. And so our standard statistical procedures might not work, and the standard error estimates may be too small. And so the doubling of the word standard on this slide is intentional because we have the standard estimators of the standard error. I said, is that a typo? No, it's not a typo. And so potential solutions to that would be to use a permutation or resampling based approach or use a small sample correction. So these are things that would have to be written into the analytic plan to ensure the validity of the resulting inference from a type 1 error perspective. So then addressing the concerns about everyone not receiving the intervention, that not being appealing due to enrollment, or the burden of rolling out the intervention to all of the sites randomized to receive it at the same time would bring us to consider a stepped wedge cluster randomized trial, which is a one-way crossover cluster randomized design in which clusters cross over from the no intervention or usual care to the novel intervention state over a number of steps. And so in this figure, we note that we have 6 clusters that are one by one crossed over from the control to the intervention state across these 7 periods. Initially, all clusters are observed in the standard or usual care state, and then all clusters at the final period are observed in the intervention state. And so each cluster will have, or the clusters will have varying numbers of periods in the standard or in the intervention states. Once clusters cross over into the intervention state, they stay there. And then the outcomes are measured at each of the steps. In this figure, we have one cluster crossing over in each period, but in other designs, you might have multiple clusters crossing over in each period just based on kind of what's logistically feasible. So some advantages for the step wedge design are that it's appealing politically since everyone sees the intervention. There's a greater feasibility of rolling out the intervention in a staggered way. And they can be more efficient than parallel RCTs for large values of intercluster correlations. And this is similar to what we observe in crossover designs, or 2x2 crossover designs at the individual level, where you're using within and between unit information in order to estimate intervention effects. And that can be more efficient than the simple parallel designs where you're relying entirely on between randomized unit information. And then because they're inherently longitudinal, they allow for the evaluation effects of time and potential interactions with time for the magnitude of the intervention effect. A big disadvantage, however, is potential confounding by time and potential secular trends, as with any type of crossover or pre-post design. On average, treated outcomes occur after control outcomes. And so if that coincides with secular trends, then we could get biased estimates of our intervention effects. There's also burdensome data collection because we're collecting so much data from all of these sites across time. And then the biggest challenge that we've run into is potential deviations from the study schedule that undermine the potential benefits of the randomized starts. So how do we power effectiveness implementation hybrid RCTs, thinking of these three designs? So again, starting with the parallel individually randomized trials, just because they're an easy starting point that some of us might be familiar with from our introductory statistics classes, right? We recall the power is the probability of rejecting the null hypothesis when it is indeed false, and we often represent it by 1 minus beta. We'll use the two-group superiority test of equality of means in a continuous setting just to motivate our concepts. And then we know that along with the power comes the consideration of the sample size, which is the number of participants that are required per these two groups for a desired level of power, right? So we see our null hypothesis and our alternative hypothesis listed. The alternative hypothesis should be that it's not equal to 0. And then the parameters that we have are the type 1 error, the desired power level, the sample size, the magnitude of effects, and the standard deviation. And we have these power expressions here that rely on normal theory, right? So we have this function of these parameters that reflects our power. And there are these trends where, as your effect size gets larger, your power is greater, and you don't need as many participants. As your variation gets bigger, then your power goes down and you need more participants, et cetera. So if we had an individually randomized trial where we had implementation and effectiveness outcomes both operating at the individual level, we can, in parallel, think about our effect sizes, and we would just note that we would have different effect sizes for the implementation and effectiveness outcomes. So we note here that we have a difference in means for the implementation outcomes with indexed by I, and a difference in means for the effectiveness outcomes indexed by E. And we would have to consult the literature and be aware of what our outcomes were and what clinically meaningful differences are for each of those in order to power our study. So we're going to get a required sample size according to each of these objectives, right? Our NI and our NE. And we're going to want to select the maximum sample size across those two so that both objectives are adequately powered, right? So we wouldn't want one objective to be adequately powered and the other one not. So we just want to be aware of selecting the sample size that will result in both being adequately powered, which means that the one that requires the smaller sample size is going to be slightly overpowered, right? But that's part of this reconciliation process of reconciling the design across the implementation and effectiveness objectives. So this is a example in R. This standard individual level inference with two groups. You can do this in pretty much any statistical software package such as Stata, SAS, PASS is one that allows you to do more flexible and more complex power and sample size calculations. So this is very accessible to clinical researchers and various researchers. For cluster randomized trials, we need some additional parameters. And we note that we have to consider sample sizes and in terms of the number of clusters, but then also in the number of individuals per cluster. And so the required number of observations per arm to achieve a certain level of power looks similar to the individually randomized case, but it has this design effect here, which is a function of the intra-cluster correlation rho and the number of individuals per cluster M. And so that creates this inflation factor where you need a greater number of total observations in a cluster randomized trial to have the same information that you would in an individually randomized trial. This design effect increases with the magnitude of the intercluster correlation and also with the number of observations per cluster. And so I think one of the counterintuitive aspects of cluster randomized trials is that you actually get less efficiency for larger clusters. You get the most efficiency when you have a lot of small clusters, with the extreme case being one individual per cluster, which is the individually randomized design. And as you add as the number of individuals per cluster increases, yes, your power goes up, but it goes up less as that M gets bigger, and it'll eventually plateau to where you're actually not getting additional power for adding additional individuals per cluster. So at some point, you will just have to add more clusters, and you won't be able to get around that. So for a hybrid design that's clustered or multi-level in both objectives, we would need intra-cluster correlation parameters for both objectives. We would consider sample size calculations at both objectives. And then we would again reconcile the two. And a useful feature for sample size calculations in cluster randomized trials is that you can very easily map using the design effect, the number of individuals that's needed in an individually randomized trial to the number of randomized units and observations per unit needed in a cluster randomized study. So if we know the number of individuals needed per group in an individually randomized trial, we can just multiply that by the design effect. And we note that product would have to equal the total number of observations in the cluster randomized trial when we take the product of the number of clusters and the number of observations per cluster, right? So this enables us to use very simple sample size calculators in order to power cluster randomized trials, which have a more complex structure, but has this very straightforward mapping. So in practice, this is what I always do. I always start with the individually randomized trial and then scale it up or down depending upon the number of clusters and number of observations per cluster. So thinking of our hybrid effectiveness implementation with varying unit levels, right? We could have one objective where we're operating at the individual level, like in Hattrick US, and another objective where we're operating at the in a clustered sense where we have observations at a lower level nested within the clusters. So an observational study that I work on, but I'm placing it within the cluster randomized trial setting just because we have these nice frameworks for thinking about power, is evaluation of a two-tiered care model for clinics that treat people living with HIV. So we can imagine we have a clinic level intervention was to implement this two-tiered model or to just do the standard of care, which would be a single tier. And then the implementation outcome would be clinic level fidelity. So the percent of patients that are receiving the correct care tier. The effectiveness outcome might be individual level viral suppression, which would be binary. So for our implementation outcome, we're operating at the clinic level. So we have a continuous outcome, which is going to reflect a comparison of means across two groups. And then we would have the standard deviation as a relevant parameter for considering continuous outcomes. But for our effectiveness outcomes, which are individual level and binary, we're not going to have a standard deviation, right? We're just going to have a proportion of or probability of being virally suppressed. For the required sample size for the implementation outcome, we're just thinking of the number of clinics, right? So our individual level, the notion of individual level observations is irrelevant because we're defining our implementation outcome at the clinic level, right? We're not thinking about fidelity of an individual patient living with HIV, but we're thinking about fidelity of the clinics in um, having a high proportion of patients that get the correct tier level. And then for our effectiveness outcomes, where we're looking at viral suppression, we at the patient level, then we have to think about how many patients are there per clinic and the total number of observations being the number of individuals across all of the clinics. And then the notion of intra-cluster correlation would only be relevant to the effectiveness outcome. So we would have to reconcile the number of clinics that are in the study with the number of observations that we expect per clinic to have adequately powered effectiveness and implementation objectives. Um, so then thinking about stepped wedge cluster randomized trials, there's even more parameters to consider. We have the number of time steps and the number of clusters crossing over per time step, the number of individuals per cluster. And because that's longitudinal, we now have to think about whether the individuals per cluster are participating in the study in a cross-sectional, repeated cross-sectional, or in a longitudinal fashion. So are we assessing the same individuals at each time within each cluster, or are we getting different individuals that are nested within each cluster at each time of assessment? And that is going to have very big implications on our power and the amount of data that we need to collect. Next, we could have multiple intra-cluster correlation parameters to characterize the correlation of individuals at the same time in the same cluster or of the same cluster but at different times. And you might think that that correlation might be lower than individuals in the same cluster at the same time. Or we can create a function of correlation that decays over time. So there are all of these additional potential complexities in the assumptions that we make when designing these stepped wedge designs that are popular in implementation research. So there are a lot of calculators that you can find in order to power a stepped wedge trial. The one that I've used is the first one on this list, which is the R-Shiny app for cluster randomized trials generally, but I often go to it when I'm trying to power a stepped wedge trial. And these are some others that I saw listed on the website Stepped Wedge Hog, which is a resource for individuals who are interested in conducting step wedge trials. Yeah. So this is what the R-Shiny calculator looks like for power and sample size for cluster randomized trials. So this is accessible online. And here you note that you're going to select your trial design. So here we've noted stepped wedge. We note the sampling structure because it's cluster randomized and because it's longitudinal, we again have to indicate whether we have a cohort, meaning the same individuals within each cluster followed over time, or cross-sectional, meaning different individuals that are assessed at each time point within the cluster. And then the correlation structure is indicated here. So exchangeable is going to imply equal correlation kind of within time and across time for individuals within the same cluster. And we often assume it because it's simple. And we don't always have all of the information that we would need in advance to define a more complex correlation structure. I think that again speaks to when studies are published, it's very useful to publish that correlation information to help people design later studies. But we could also incorporate some type of decay in correlation for designing a stepped wedge trial. So here we see the power as a function of the number of clusters per sequence. And then here we observe additional display options. So you can display the power and sample size considerations as a function of fixing the power and varying the number of clusters versus the cluster size, or as fixing the cluster size and looking at the number of clusters versus the power. So here, this shows fixing the power, but varying the cluster size and number of clusters. And so you see as the number of clusters increases, the cluster size goes down for a fixed power. We have to specify the power and we have to specify the number of steps or the number of longitudinal periods over which outcomes will be assessed. Right, so this is how this calculator is used. It's very, it's simple, but you do have to kind of understand the elements that go into it. So then let us talk about multiplicity. So this was something that, in every field, I kind of ask, how do people typically think about multiplicity and do they tend to adjust for multiple comparisons? And so my experience so far in this hybrid implementation effectiveness world is that we don't, I think historically, effectiveness trials and implementation trials were different things. And when we're bringing them together within the hybrid designs, we're still very much conceptualizing of them as different studies. And so we don't typically adjust the type 1 error rate for the fact that there's two outcomes when we have a single effectiveness outcome and a single implementation outcome. But if we were to have multiple implementation primary outcomes or multiple effectiveness primary outcomes, then we should adjust the type 1 error rate to account for multiple comparisons within effectiveness or within implementation. So some concluding remarks. Hybrid effectiveness implementation studies are kind of two studies overlapped into one, and the design elements need to be considered separately for these two objectives. And these implementation effectiveness objectives may operate at different levels that require us to think about sample size a little bit differently. And then some challenges that we've experienced and some lessons learned. The biggest one is just to anticipate time delays. Everything takes longer than you would expect. Data acquisition, contacting people to initiate rollout of the intervention. So start early. Relevant to the stepped wedge trials, adherence to the study schedule has been really challenging, just as far as getting all of the participants together in order to start the intervention process and to start the tailoring of the protocol at each of the sites. And so when thinking about whether you want to use a step wedge trial in your implementation research, think about the logistics of the intervention rollout and whether it requires a lot of coordination because that across a lot of different people, because that can lead to delays that really challenge adherence to the randomized study schedule. One of the stepped wedge trials that I am on the DSMB for, it was more of a pilot. It had maybe 4 observations, but they just said the stepped wedge is out the window and sites will start when they're ready. And because this is a pilot, that's okay. But if we want to draw more rigorous conclusions about the effectiveness or about implementation, then we would be really concerned about bias associated with that because it might be that the better sites are starting earlier. And so yeah, there are a lot of problems or challenges with adhering to the study schedule. And you really want to think about the logistics of your intervention and outcomes to see if a step wedge trial is likely to work well. And then I think another challenge that we've observed is just data acquisition and harmonization across the sites. And I think, Megan, when I write that, I'm thinking particularly of these patient safety indicators and these kind of regulatory outcomes that are submitted for quality, but maybe aren't research ready. And so I think just time delays in dealing with those and really having to dig into various data sources and harmonizing them across the sites is going to be a challenge. So I will stop there and then we can have some discussion. Thank you all.
Alisa, that was great. Thank you. And I'm really glad that we have you on our team because I followed all the variables, but I can't say I understood everything. I understood conceptually, but I'm glad she does that. It's
Thank
important
you.
that it's a team science.
Yes.
And for those of you who have not done implementation trials before, it's really important to have a colleague with biostatistical expertise, but specifically one who understands implementation science because it is different. And having someone who can sort of understand the nuances between the two different study types is really important.
One question that we had gotten from a participant ahead of time that we wanted to answer now, they had asked, how do you elevate and differentiate research from quality improvement in the context of wanting to do an implementation science research study?
Oh, that's an interesting question. I mean, we could talk about the philosophical differences between QI and implementation science. I'm increasingly thinking it's not so useful to do that, but there's overlap in terms of approaches. In general, quality improvement is focused on local change that's really timely, and implementation science is focused on building generalizable knowledge. So even though the some of the approaches may overlap, the objectives tend to be different, and so then the judgment calls that you make tend to be different because you're trying to create that generalizable knowledge in implementation science. You need to pay a lot of attention to theory and rigor and precision and measurement, whereas in QI, it's not that those are unimportant, but sometimes there's this concept of good enough, where you want to learn enough about something to keep it moving because you really want to help the folks that need help right now. Um, in terms of describing studies, it can be sometimes useful to describe an implementation study as like quality improvement. And we've certainly done that with the Hattrick study, where you go into an ICU and then you say you're a researcher and they're like, what do you want? And it's like, well, no, this is a lot like quality improvement because we're trying to get people to communicate better. And they go, oh, okay, that makes sense, right? So when you're trying to engage participants, drawing parallels to quality improvement can be super helpful to give them a sense of what is happening. So when you say research to a lot of people, they're thinking like drugs and syringes and like, that kind of thing. Um, so that's I found it useful.
We have a question in the chat about intuition for stepped wedge trials and power. When observations within clusters are observed in multiple time periods or when it's repeated cross-section. So this depends on the kind of within individual ICC. So in a cohort study where we have the same individuals that are observed within a cluster over time, um, if that correlation is high, such that the pre-post comparison uses that within individual within individual correlation, then that is going to kind of reduce the variance associated with our estimation. So there's not necessarily a generalizable rule for if the cross-sectional or cohort approach is more efficient. I think it depends on whether individual level outcomes are highly correlated for the same individual repeated over time. If there's not that high correlation of individuals over time, then the repeated cross-sectional might be more powerful. And I think that's the same trend or pattern that we observe in like 2x2 crossover trials, where you have to have enough kind of within person correlation across the repeated measures for that within individual information to help with efficiency and to be more efficient than using the between person information. Oftentimes it is, right? Because people tend to be highly correlated with themselves.
Other questions about study design? If any of you have had experience with stepped wedge trials and you want to talk about it, I know Rebecca's running one right now. I'm curious if you have any insights for folks. And maybe you can tell them a little bit about your study.
Sure. Um, Alisa is also the savior of my study as well. Uh, we are doing a cluster randomized, well, a randomized rollout stepped wedge trial, where we have 14 labor and delivery units across the country. They are all going to be implementing a calculator that predicts likelihood of C-section at the start of labor induction as a part of their clinical care, for all patients who sort of meet eligibility for that. So they are randomized to start times 10 weeks apart across a 3-year period. So everyone has a different, just like all of these ones that we're showing, a different amount of pre and post period. And it is something where we are still at the very beginning. So the beginning of that 3-year period started this September, and our first site implemented just this week on Monday on time, which was a possible miracle. And so, I am sure that that will not stay the case throughout the time. But I think that one of the things that, is a little bit nice about the intervention I'm utilizing in this space is that it is something that can be released in the EMR, in the electronic medical record, at a given time point if it is ready to be released in the EMR, which is not, always the simplest thing, and that each of these sites needed to, incorporate the calculator into their own EMR. And I only had so much control over being able to do that. Different processes work very differently at different sites in regards to their EMRs. I'm sure you guys all know. And so, yes, so, we can turn it on at a given time point if that is, if it's ready to do so. And so that is the plan. But certainly we expect that some places will have to implement without their EMR being ready. And then the EMR part's going to be turned on later. And Elisa's already thought about how we're going to statistically incorporate that into our analysis. And that there may be some strategies that kind of go on and off over time. And so there will be complexities to that as well, but it is in the maternal health space in this, and has a similar study design to Hattrick. A lot of it was modeled off of the Hattrick trial.
Awesome, thank you. All right, any questions before we draw to a close? We're getting close to the top of the hour. So I posted in the chat, feel free once we post this recording to share it with your biostats colleagues, because, as you bring them onto your studies, they may have some questions, and this content will be probably particularly interesting to them. Dr. Stephens Shields, it was amazing. Thank you for sharing your brilliance with us.
Thank you for
And
having me. I really appreciate it.